So another technique is what we want to see. So that is 10 microns. That is 2D. Right? We want to see 3D. So how to do 3D? So now we try this method now called clarity to make the brain transparent. Because if the brain is opaque, like a normal brain, the light cannot. We can't use the light. The microscope light doesn't penetrate. So we can't do it. But then if we clear the brain somewhat, then we are able to do a 3-dimension. So this is what you can see, the exercise, right, which is wrapping the processes around the blood vessels. So this hole is the blood vessels. It's a huge blood vessels. So the technique what we did is these are the two, and I need to acknowledge these two people who actually do quite a bit of the work. This is what the recent publication we have in this journal. So we use a technique that to study glioma. So what we do is we implant mouse glioma cells into the brain of the mouse brain. So and we treat them or give them or the mouse maybe under different genetic background. So we use that, and then we use do the sectioning as well we've done before. So this one, this picture, what you see, the blue thing is actually the tumor cells, and you can see the red. The red are all the exercise, and they are trying to constrain the tumor cell. So they are forming a border. So this is a protein we are interested in, the green. Now it forms yellow when it overlaps with the red. So the green is the protein. You can see that it's not expressed anywhere else. It's only expressed around the tumor. So while we are speculating that it is actually doing something, right? It's definitely what if. So what is the role? Is it trying to help the brain? Is it trying to stop the tumor cells from invading, or what? So this is what they really want to do. So another thing is that we also notice that, so this is another injury, which is a stroke, and you can see that using the again, PDBA is just one of the stem cell markers that we're interested in, and you can see just very close to the border. So this is what I was saying before, that during injury there's activation of stem cells, and now we prove that this is also in the brain, and this actually has great ramification, because what does that mean? Can the stem cells actually form neurons back, or actually they make things worse, right? Because they form a scar that the brain cells cannot cross over to the uninjured side. This is just to show you again, really what we are interested in is the immediate neighborhood where the tumor interact with the normal brain. So basically, this is just the case. This is what we do, what we are interested in for the neighborhood. So now this is what a typical cell biology signaling transduction lab will do with animal model. So for example, right now what we are trying to do is, you can see that the blood vessels, which is now stained red, and the tumor cells are green. You can see that in the tumor, the blood vessels are large, huge compared to the blood vessels outside. So the tumor cells actually try to encourage the blood vessels to grow larger, so that they can get more nutrients and they grow faster. So this is how cancer cells really try to hijack the normal brain function so that it can survive better.